Hello everyone and welcome back to microbiology. Today we'll be covering the Unit 3 practice review for the exam. We'll go through these pretty quickly through the lecture, but as you work on your own, please feel free to pause each one of these slides and take your time with the answers. Again, the way this works is I'll present a term or a concept, and then you're going to try and regurgitate as many facts as you can or concepts about that particular topic. Okay, our first one is ribosomal subunits of the prokaryotes versus eukaryotes. Okay, so as a review, the ribosomes are the protein synthesis factories of the cell. For prokaryotes, they produce a 50S or Svedberg subunit and 30S Svedberg subunit, and the total does not equal the sum. The Svedberg unit of the two subunits combined together as a whole is 70 subunits, so it's a 70S total. And the eukaryotic ribosome is made up of a 60 and 40, and its total is an 80S. These ribosomal machinery complexes are important because depending on their structure and their function, they can be inhibited by antibiotics differently. The next one is tetracycline. Okay, tetracycline is an antibiotic that blocks the 50S subunit. What it does is it actually interferes with the docking site of tRNA, and without the tRNA, it cannot release the amino acid according to the anticodon. So in this image, the little LL here represents the tRNA, and because this one cannot dock in the next codon region, it won't donate this amino acid. And this is only going to be effective on the 50S subunit. However, mitochondria do have a 50S subunit, so this would be somewhat toxic to eukaryotic cells as well. The next one is Acyclovir. Acyclovir, also known as Zovirax, actually is a synthetic version of guanosine, which is one of the 4 DNA nucleotides. As you can see here, the relationship between guanosine and acyclovir is nearly identical. The ribose sugar, however, is altered. And because of this, it can mimic guanosine, and this causes terminations of new strands of DNA when it gets incorporated into DNA synthesis. And this has selective toxicity because it requires an activation by a viral enzyme. So no viral infection of the cell and no assimilation of the guanosine, of the acyclovir. Okay, azidothymidine. Okay. Azidothymidine, also known as zidovudine, is a compound that mimics thymidine, as the name sort of insinuates, only has this amine group here that distinguishes it. And what this does is this inhibits the enzyme reverse transcriptase, which is used by certain retroviruses. And so this would do something like prevent HIV retrovirus converting its RNA into DNA, and therefore integration into the host genome cannot take place. Next one is Ivermectin. Ivermectin blocks the neuron transmissions in helminths, which causes paralysis and death. Here we have target modification as a mechanism of antibiotic resistance. Target modification is one of the 4 types of antibiotic resistance that we see in microbes. Target modification involves changing the structure of the thing on which the drug acts, so therefore the drug can't bind to it. So if you alter maybe a site at which a amino acid attacks, or sorry, an enzyme attacks, then the enzyme won't recognize that site. If a amino acid base change happens, an amino acid change, let's say, happens in the ribosome, then maybe something like tetracycline cannot interfere with the docking site of that ribosome. So we can see how modification of the target for which the drug acts can lead to circumvention of its toxicity. Prodromal period. The prodromal period is the second stage in an illness, and it is a short interval where incubation of some diseases occurs, and it has early or mild symptoms which are experienced, and the number of microbes begins to increase in this stage. Exotoxin. Okay, so there are both endotoxins and exotoxins. Endotoxins is probably the most difficult concept to grasp because you can have compounds that might exist on the cell wall, which seems like the outside of a cell. And when that cell dies or divides, it can then get released from that microorganism. So a lot of people confuse that with being an exotoxin. But anything that's a component of the cell itself is an endotoxin. Exotoxins are much different. They're mostly produced by gram-positive bacteria as a part of their metabolism, and they produce specific damage to host cells, whereas endotoxins just cause general inflammation. Okay, next one is antigenic variation. Antigenic variation is where some microbes can change their antigens before the host can mount an immune response. These are slow changes over time. Here we can see that small changes in the antigens shown here in these curves lead to the requirement of new antibodies that can identify those antigens. Okay, vector transmission. Vectors are animals that carry pathogens from one host to another. Very common vectors in human diseases are insects. We have 2 different types. There is the mechanical transmission, which involves passively transferring diseases through the body parts, such as this fly landing on this hamburger. Or we can have biological transmission, where this is actively transmitted via a bite from an insect, carrying it from one host to another. Next is living reservoir. So diseases can be transmitted in 2 ways, from a living organism or an inanimate source, and these sources are called the reservoirs. A living reservoir can include humans, which are both affected and carriers, or through wild and domestic animals. Next up is parenteral route. Parenteral route is one of the 3 portals of entries. It's often confused with mucous membrane or skin entries. That's because it can occur on either of these surfaces, but the way that the organism gains access to the host is through a breach. And so this would be a sore or a wound. Next is pandemic. So we can have different ways to describe a disease. It can be sporadic, where it occurs only occasionally in a region, such as the plague. It can be endemic, which means that it's constantly present in a region, such as malaria in sub-Saharan Africa. It can be an epidemic, where it's present in a large number of people in a region. And it can be a pandemic, which means that it's present in a large number of people worldwide. Now we have indirect contact transmission. Okay, so there is direct contact transmission, where the pathogen is transmitted through direct physical contact between the two hosts, and so there's no sort of fomite or physical intermediate between the two. There can also be congenital transmission, where the pathogen is transmitted from the mother to the fetus. An example of this is HIV. And then we have indirect contact transmission, which is what we just described direct contact transmission not being, which means that there's some sort of an intermediate inanimate source, such as a fomite. And we can also have droplet transmission, where the pathogen is spread in a short distance, mucus droplets created through coughing, sneezing, talking, etc. Next up is lacrimal apparatus. Okay, so in this image here, we can see a skin structure where you have the epidermis and the dermis. These are for skin and mucous membranes. We also have something called the lacrimal apparatus, and this is where tear ducts are for draining tears. And I won't go through all the rest of these here. Let's just go to the next term, which is interferons. Interferons are proteins that are produced by virus-infected cells that stimulate phagocytes. Okay, next one is heavy chains. This is of the antibody, if you'll remember. Okay, so here we have an image of an antibody, and the heavy chains are colored here with blue and an H, so we can see there's a heavy chain on each branch of the antibody. And down here at the stem, there are two identical light chains on either side of the branch and two identical heavy chains. The stem is called the effector constant, or FC region, and classes of antibodies are distinguished based upon this region. One other thing that you need to be familiar with is that the epitopes are identified here at the antigen binding site at the end. Okay, tell me about the IgG antibody. All right, here is what we need to know about the IgG antibody. This is the table 17.2 summary. First, its structure, it's a monomer. It's not a pentamer or a dimer like IgM or IgA. It is the major constituent of the total serum. It's 80% of all antibodies found in the body. It can go through placental transfer, and it is useful in protecting the fetus and newborns. And, you know, there's a whole lot of information to digest here. You need to be able to pick out what's unique, and what I've sort of circled in red here is going to be most important to you. So some of these structures are important to know. The percent of the total antibody for IgG is important. You need to know that's the dominant one there. you need to know where they occur. So for example, the IgA has this secretory component, so it can be secreted into your saliva, mucus, intestines, milk, and it is also present in the blood and lymph naturally. So yeah, you just need to focus on what's really important here. These very small amounts of total serums for these other ones aren't so important. The molecular mass, again, not so important. Just focus on kind of the more important things that I've circled in red here for the exam. Okay, now let's cover adaptive immunity, natural/artificial, and active/passive. So adaptive immunity can occur in two ways. It can occur naturally, where you're exposed to the pathogen or your mother is, or artificially, where it's acquired through a medical guidance from a medical professional. By that, what I mean is you can be vaccinated or be given antibodies. We can break these down even further into active and passive. Active is when you gain the ability to produce these antibodies and specialized lymphocytes yourself, so that you're capable of having long-term immunity. But passive immunity occurs when you receive those antibodies from your mother via the placenta or via milk. We have active and passive also for vaccinations and antibody therapies. So you can be given antigens from a virus and then produce an immunogenic response and have your own antibodies and specialized lymphocytes, or you can just be given the antibodies from somebody else. And this will hopefully give you some sort of immunity to that pathogen. Next up is systemic anaphylaxis. So systemic anaphylaxis is an entire body reaction, an immune reaction. So your blood vessels throughout the body dilate, and as a result, the fluids escape your blood, and it causes your blood pressure to drop, and you go into shock. Airway passages begin to narrow. You can have a skin rash, nausea, tingling sensation. This can be fatal within minutes, and it's typically treated with an injection dose of epinephrine to constrict blood vessels. Okay, types of hypersensitivity. So this is one of those really information-dense tables that you're going to have to digest for the exam. There's a lot going on here. We have type 1, which is an anaphylactic reaction, type 2, which is cytotoxic, type 3, which is an immune complex, and type 4, which is a delayed cell-mediated or delayed hypersensitivity. I circled in red basically the important things to recall for the exam here. So for type 1, or an anaphylactic shock, this is pretty quick. It takes less than 30 minutes to show signs, and this can involve IgE, which binds to mast cells or basophils and causes the release of histamine. And as you can imagine, with this really quick reaction, this is very common for allergic conditions. Type 2 is cytotoxic. So cytotoxic, we're dealing with a target cell here, right? Cyto for cell. It takes about 5 to 12 hours. The antigen causes formation of IgM and IgG antibodies that bind to that target cell. This can occur with, as you can imagine, with target cells, transfusion reactions, or the RH incompatibility that can occur. Okay, the next one is type 3, or immune complex. This takes about 3 to 8 hours, so there's a little bit of an overlap with type 2 cytotoxic reactions. This involves antibodies and antigens that come together and they start to form these larger complexes that lead to damaging inflammation. Type 4, or delayed cell-mediated or delayed hypersensitivity, takes a while longer, right? It's delayed 24 to 48 hours. So the antigens end up activating the CTLs that kill the target cells. And this can happen with transplanted tissues or other contacts with things such as poison ivy. And again, for the exam, I want you just to pay attention to the things in red here. Of course, you're going to have to, I didn't circle the names for the types of reactions. You'll need to know those, but the information you need to know for those are circled in red on this table. Okay, tell me about characteristics of plasmids. Okay, here are the characteristics you need to know. They're a circular piece of DNA. They're also sometimes called vectors. They are extrachromosomal. They aren't part of the chromosomal DNA, and they contain accessory genes such as antibiotic resistance. They can be exchanged during conjugation through horizontal gene transfer, and they can be inserted or transformed into cells in molecular cloning. Okay, steps in traditional cloning. Here are the very basic steps to this. The first thing we need to do is we need to get our gene of interest and amplify it through PCR. After we've amplified that, we need to digest it with restriction enzymes and digest the plasmid we want to insert it into. And so now they both have digested ends that match that same address. And because of that, they can bind to each other, and we can seal up the broken phosphodiester bonds with a ligation in step 3. After that, we can then do our transformation and then our screening or plating on antibiotic plates. So that wraps up the review game for Unit 3. I would pay very close attention to what I circled in red in a couple of those tables. If any of you have any questions, feel free to contact me with those questions. Anyway, good luck, study hard. Bye-bye.